Welcome, Life Sciences. Welcome to another exciting life science lesson. What are we starting with today? We're starting with an extremely important, section of the work, and that is going to be cells. When you had a look at the biochemistry section, you would have had a look at all the inorganic and the organic nutrients, and we're now going to kind of put those together to come up with this concept of what a cell is. Now, I posed an interesting question on the board. If you have a look here, I wonder if your mind can actually take in the volume of what I'm talking about. And I've put that on the board. Have you ever wondered how many cells are in your body? And I Googled it, and generally the answer that I came up with was 37.2 trillion. Now, I'm not a maths person, but I just know there's a heck of a lot of noughts that are going on to go to that thing. And that is what you're made of. All these things called cells. So we're going to look at this whole concept of what is a cell? And remember, when we're looking at a cell, we're going to build up further what is a tissue, what is an organ, what is a system. So today we're going to lay down the foundation of a lot of work that we're going to look at later. Now, when we look at this concept of the cell, we're going to do the general concept. We're also going to look how things move in and out of a cell. So there's going to be a lot of terminology during this section. So as I always say to you guys, please make sure that you go and have a look at these terms. I think they're going to be very new to you. You might never have heard them before. And sometimes you might also battle to pronounce them, to say them. So try and go over it. Go and look in your textbooks. Make sure that you understand what this concept is that we're looking at. Now, when we look at what a cell is, when we look at all living things, I always teach my grades, there's a little acronym that I look at, and it's called, Mrs. Some people say Gref, or some people say Gren, right? And here it means that all living things must be able to move, cellular respiration. They must be sensitive. That doesn't mean that they cry. That just means that they are sensitive to things around them. All living things must be able to grow, to reproduce, to excrete, get rid of wastes, and to feed. So it's a nice little acronym that I use there. And when we're looking at this living things, all living things are made up of this thing called a cell. And that's living things. So I've put here a fun little picture, but it's plants and animals. And there's even stuff that we can't even see, that we're going to consider as living. Now, when it comes to the study of cells, you need to understand that it's become a technology. What scientists and biologists used to understand about cells is, if I could say, a fairly recent concept, And why I say recent is it's all about technology. And the technology I'm talking about is the microscope. We have now been able to get such a greater understanding of these really, 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 really small things because technology has allowed us to do so. Okay, so what am I talking about? When we talk about the microscope, I don't know, maybe some of you in your classes are privileged enough to have a light microscope, I know I'm lucky enough, I have a few of these in my classroom, and my kids in my class are able to have a look at cells. Now, the reason why I put this one up is because it's the first thing, the practical I do with my children in my class. I get them to have a look at an onion cell. Now, if you're having a look at what that is, I'm going to show you. We're going to look at the following things when we get to a cell. A cell has got something that's around it, something that's in it, and a very important something that's in it. And that we're going to have a look at the terminology just now. So if you're lucky enough, you would have had a light microscope, light emitting microscope. Now, the ones that we're going to find at the universities and all these places, right? This big, right, microscope is called a transmission electron microscope. And what it enables us to see is often what we call a micrograph. Now, what of the pictures I've put up over here? There we were able to see just a simple cell. This one, this is an animal cell, and this is a plant cell, and we are able to see such great detail because of this microscope. Very often, right, maybe in your tests or in your textbook, they're going to show you what the things we look at. We're going to give you a diagram, and very often we're going to show you what it actually looks like in real life. Then the last microscope that we're looking at is called a transmission electron microscope. And if you have a look over here, okay, this is a microscope that shows us organisms in 3D. Now, these organisms that we are showing here, right, are part of COVID, right? The SARS-CoV-2, the virus that is, right, the pandemic that we have. And what we can see is a virus is something extremely, extremely small. And this machine allows us to have a picture of what the virus looks like in 3D, right? What it gives you an idea of its shape, of its structure, etc. So what we've noticed through time is because of the technologies that we now have, we are able to see, all right, these really, really tiny structures in a great amount of detail. Now, when it comes to a cell, there are two types of cells that we find. Now, the first one is what we call a prokaryotic cell. And if we were to put it in a timeline, these are the oldest living cells. Now, when we look here, the prokaryote is a single cell, a unicellular cell, and falling under this category is going to be bacteria. They are the oldest living thing. And what we find is that they were very, if I could use the word simple. They had a simple structure. They had something around them. All right, we're going to look later at this concept of a cell boundary. They had this jelly-like substance inside it, right? And then they had their DNA, because we were looking at this whole concept of reproduction, carrying on our genetic material. But when it came to living things, they were very, what we call simple. Now, if we have a look at, when we look at classification, all right, later on, organisms over time have shown a pattern of becoming more complex. Simple in the beginning, and then as time has gone by, so they've become more complex. And that is the same that's happened with living things. Bacteria was the first living thing, all right, on Earth. And as time went by, we saw more complex characteristics in organisms, both plants and animals. Now, with that complexity, let's have a look here. We find a cell, it's called a eukaryote. And what it basically means is this cell can do more complex things. Now, I know this sounds like a bit of a mystery, but as we go through the whole structure, you're going to see what I mean. What it simply means is there are more things, we call them organelles, in these cells, and they are able to do more. It doesn't mean that they are better. It just means they are able to do more things. And we are going to concentrate in this section on the eukaryotic cells. You will look at prokaryotic cells a bit later. Then what we also look at is the following, that eukaryotic cells, right, can be divided into an animal cell and a plant cell. So we're going to find that plants and animals have got different structures because sometimes they need different things. They have a lot in common, all right? There are things that they have that are the same, but a plant and an animal do things differently, so they must also have things that are different. Right, so when we look at the end here, we need to be able to see, is it a plant, is it an animal, and why do we need to know that? The last concept that we need to look at is this concept of what is unicellular and what is multicellular. Now, uni means one. And when we talk about one, you need to understand the concept that a single cell, one cell, can be living. There's an amoeba, there is bacteria, there's some protists. One single cell is enough for it to be a living organism. It undergoes all those concepts of Mrs. Wren. However, again, what did we know? Things usually went from simple, all right, to more complex. And with that complexity, we made organisms that are multicellular like us. We've got lots of cells that are put together because they need to do more. All right, guys, so with those basic concepts behind us, we're going to take a quick little break and then we're going to be right back. Welcome back, Life Sciences. I hope you had a good break. As I always say, got to get up, got to get those muscles working, got to get the blood flowing and the oxygen flowing. What are we looking at today? We're concentrating on this concept of a cell. Now, when we look at the cell, I want you to picture, we're going to, just for the beginning, to picture one small, very small little thing that we're going to look at, right? And then we're going to look at all the differences and the importances from there. So when I'm looking at a cell, what am I looking at? Okay, so here I have a structure in front of me. Now, all living cells have this basic concept in them. Right, now the first thing that we're going to look at is this concept of what cytoplasm is. Now, I want you to think of the following, all right? Let's make jelly. Why am I asking that? Because that is what the cytoplasm is. I want you to get a bowl, all right? So picture a bowl. The bowl would be the outside enclosure called the cell membrane, but we're going to get back to that. Inside this bowl, all right, so let's make jelly. I take a packet of jelly, and it's got all dried stuff in it. What do I mean by dried stuff? There's sugars, right? But all of it is dry. So it's a whole lot of things. So if we have a look at biochemistry, that we looked at the molecule, that whole concept of, right, all organic and inorganic, it's pretty much, it's got sugars, it's got minerals, it's got maybe vitamins in. And how do I make the jelly? What is the most important part of the jelly? Is the water. Because what do all living things mostly consist of? Water. So imagine now I take this bowl, I put my dry contents in, and I put the water in, and I stir it. That's exactly what the cytoplasm is. And why did I use the jelly? Because that's pretty much the concept of a cell. It's neither a solid nor it's a liquid. We use the word sol and gel, right? Sol more liquid, gel more solid. But that's what the cytoplasm is. Remember, when you eat this food and everything that you eat, where do I store it? I store it in the cell. Dissolves in the water, and it's there in the cell when I need it. Okay, so the concept of the cytoplasm, all right, is this bowl of jelly. And as I said to you, it's got the most important thing is this water. And this is what it is. All of this over here, right, is water. And then in the water, I'm going to have some gases. What are my gases? Oxygen and carbon dioxide. And I'm going to have some minerals. What are my minerals? What did I look at during biochemistry? I looked at calcium. I looked at sodium. I looked at phosphates. I looked at nitrogen. All right. All of my different minerals are there waiting in the cell. What else is in the cell? All right. I've got proteins. I've got carbohydrates. I've got fats. All of these are inside my cytoplasm. Now, when we look at what the cytoplasm is, you need to understand that not all of my jelly, all right, all of my things are dissolved. Now, imagine you take jelly and you put, I'm going to go weird, put a jelly bean in, all right? Oh, now what's happening? The jelly bean is going to be there, but it's going to be held in suspension. Right, what happens if I put some fats into my jelly? Not very nice, I know, but we know fat and water don't mix. So maybe it lines at the top, maybe it lines at the bottom, right? It's this whole emulsification. So this jelly is holding things that are going to be inside the cell. And some of it might be my carbohydrates that are dissolved, or maybe it's got proteins or lipids that are just hanging around. Okay, so we got this concept of what the cytoplasm is. All living things have got the cytoplasm, all of them. Very important that you understand that concept. Now, the next thing we are going to look at is the cell membrane. Now, the cell membrane, all living cells have this membrane around. If I made my jelly, I had to make it in a bowl, because everything has to be inside the bowl. And that's exactly what my cell membrane does, right? It has to separate the inner and the outer. What do I mean by that? Is that a cell keeps things in, right, where it should be, and it keeps things out. So there are things that should be in the cell, and there are things that should be outside the cell. And what controls that is the cell membrane. It's like putting a fence around your yard. What should be yours is in, and what is not yours might be on the outside. Now, very important concept as well. Guys, it's the cell membrane that controls what enters and exits the cell. The cell membrane will decide what comes in and what comes out. And we use the term, there's so many of them. I prefer using the term selectively permeable because it's easier for me to spell than differentially permeable. What does selectively mean? If you select, you choose what you want to come in and out. Okay, now you don't have to know this, all right, this diagram off by heart, but you have to understand the concept of what the cell membrane is. Now, when we have a look at the cell membrane, we very often talk about the fact that it is a fluid mosaic model. Now, if I had to break down the words, right, what do I mean by fluid and what do I mean by mosaic? Okay, so if I take fluid, it means that the cell membrane can move. Now, that is quite important. A cell membrane needs to be able to move. Why would that be important? Have a look. What happens if I'm an animal cell? What do animal cells need to be able to do? If I'm just one single animal cell, I'm an animal cell and I need to eat, which means I need to hunt, which means I need to move. All right, so when it comes to my outer covering, if my outer covering was hard and I couldn't do anything, I'd die because I couldn't eat. So I've got, what have I got? I've got the outside that can move, and what is my inside? It's jelly. What can jelly also do? It can move with me. Okay, so first of all, most important, fluid, it is this, all right, outer covering that is flexible, if I were to use that word. Now, what is a mosaic? When I make a mosaic, I put pieces together. Now, I want you to notice, as I said, you don't have to notice all the things, but let's have a look at the words here. Protein, carbohydrate, and protein, and we're going to come to this one just now, lipids, biochemistry. All of these things, when I eat a protein, a carbohydrate, and fat, why is it necessary to have these in my diet? Because they make up the basic structure of my cell membrane. Guys, the cell membrane is made up of these proteins, these carbohydrates, and these fats. And I put them all together, all right? I put them all together. That's what a mosaic is. A mosaic is putting all these different parts together, right, to come up with a structure. Now, what we're going to do is, what we need to do is that the cell, things need to move in and out of a cell, right? So we've got to have a way in which things can move in and out. But have a look at the basic concept of the majority of the cell. The cell, let me go back here. These little yellow heads over here, these lipids. The cell membrane consists of all these, what we call a phospholipid bilayer. What does that mean? They're made up of a phosphate and a lipid. There we go. What does bi mean? 2 layers. Top and a bottom. Actually, an inner and an outer. And what do we notice? That the top, the heads, are hydrophilic. What does hydrophilic mean? Like water. And we want them to like water because what is around here? There's a lot of water around here. This is also going to like water. But what do we notice about the middle of my membrane? It is hydrophobic. It repels water. And why do you think we would want that? Quite simply, we would want that so that when things move across this membrane, I actually want to make sure that they move across certain parts, can you see where those little blue lipids are, and not move through the other ones. So it's basically like a toll gate, all right, that you're going to have. So when we look at this whole concept of the cell membrane, it's essential for you to understand, right, as I said, you don't need to know it, but it's going to be this fluid mosaic molecule, and that it wants to actually, anything that it moves, it wants to put it through these channels. Right, and as I said to you, what is the function of the cell membrane? It's going to choose what comes in and out, selectively permeable. That's very important when we look at the transport. So if we have a look here, just quickly having a look, all right, just remember, it encloses the cell, and this very important thing that we're going to look at just now, selectively permeable membrane. Welcome back, Life Sciences, from that break. I know, as I say, predictability. What am I going to say to you? Yes, get the muscles moving, get the blood flowing, and let the oxygen get through the body. What are we looking at today? We're looking at this concept of what a cell is. And we saw that all cells have basic components. The first one we looked at was a cell membrane, which we're going to carry on now, and then they jelly inside the cytoplasm. Now we're going to continue looking at that cell membrane because it's going to be very important in how a cell moves things around the body. Now, if you have a look at the definitions on the board, this is quite a definition heavy, if I could say to you, concept that we're going to have a look at now. Now, remember when it comes to plants and it comes to animals, things need to move around, right? Plants need to move things around, and so do animals. And generally, when we want to move something around, we want to make it as easy as possible. And I'm going to explain to you this whole concept of what a concentration gradient is and how we're going to move things. Now, remember, when I often use this analogy in class is I choose a pupil and I say to them, I want to move you. Now, I have two options. I can move you, pushing you up the hill, right, which means I'm going to sweat, and I don't do sweat. Or you're at the top of the hill, and I can just give you a slight nudge, and you're going to roll down. That is going to be the easiest way for me. Now, what do I mean by that? I'm talking about a concentration gradient. Lots of things at the top, a very little amount of things at the bottom, and things will move from a high to a low concentration. I'm sure some of you have sprayed deodorant, and you spray deodorant in your room, and you walk out, and the next minute the whole house sprays like deodorant. Why? Because those molecules have moved from a high concentration to a low concentration, and then the whole house can smell it. And that is pretty much what we're looking at today. Okay, so let's look at this diagram again. Right, this is my cell membrane. And what's going to happen is, remember, it is selectively permeable. What does that mean? It is going to decide what moves in and what moves out. Now, when it comes to movement, right, the first type of movement is called diffusion. And you'll see I have, right, a little picture that was going over there. And what is diffusion? It is the movement of a gas, a liquid, or a solid, all right? And it moves from a high concentration to a low concentration until equilibrium is reached. Okay, so let's use this analogy. You have a cup of tea, and you put sugar in it, and you just leave it, okay? So if I sip the top part, it's not very sweet. But if I wait a little while, what will happen is the top part will also become sweet. Because what has happened, the sugar has started to dissolve, and the sugar molecules have moved from a high concentration, right, at the bottom to a low concentration at the top. Why do we stir? We stir the sugar just to make that process so much quicker, rather than me going, okay, let me wait, just now my sugar, I'll taste it. So I stir it so I can get all my sugar to dissolve equally around here. Okay, so I'm going to go back one slide so that you can see the movement again. Have a look at the bottom, all right? It moves from a high concentration to a low concentration. That's quite important for us, and that's how gases are going to move through my body. When I breathe in, oxygen is going to move via diffusion from a high concentration in my lungs to a low concentration in my body. Now, what our next concept is quite a challenging one, and it's got a few words that you need to understand. Okay. So when I talk about a solution, I want you to think about your tea again. A solution is composed of a solvent, something we dissolve in, and I want you to think here of water, all right? And a solute is what we dissolve in. I want you to think of sugar. Here we go. So I've got water, which is my tea, okay? And sugar is what I dissolve in it. Now, I'm going to get three types of solutions. Now, I want you to have a look. The blue and the yellow dots are quite important. Over here, all right, the blue and the yellow dots are the same. There are the same amount of blue, which is going to be my water, as there is yellow, which is going to be my sugar. If we have a look at the next one over here, I'm going to see that there is less water and more sugar. those of you who like 4 or 5 teaspoons in, all right, your tea. This diagram over here, what do we notice? We've got more water and we've got less sugar. Now you're asking me, what on earth does this mean? Okay. Now the name of this solution where the water and the sugar is equal is isotonic. All right, iso meaning equal. Now, these two are the important ones. This one over here has got less water, but it's got lots of goodies in it, lots of sugar. Okay, and what shops sell lots and lots of things? The hypermarket. So we call this solution hypertonic. These are you guys who like your 50 million thousand or your 3, 4, 5 sugars in your tea. Here are the guys who like maybe half a sugar. So you've got less dissolved substance, but you've got lots and lots of water. All right. You are known as a hypotonic solution. Now, what am I actually talking about here? I'm talking about a process called osmosis. And I'm going to show you what it means just now. But basically, guys, I want you to listen. Water will always move from where? High concentration to a low concentration. So if here, for example, if the cell has got a lot of water, where will the water move? Out of the cell. All right? Where will it move to? A cell where there is less water. So the word that we use here, a cell with lots and lots and lots of water, has what we call a high water potential. And what does that mean? It wants to get rid of that water. Okay, so a high water potential to a low water potential. So water will always move from a high concentration to a low concentration. So guys, very important. What is osmosis? Now have a look. It's the movement of water, very important. Now, where does that water move across? It must go across a membrane. Diffusion doesn't have to go past a membrane, but it must move from an area of high water potential, lots of water, to an area of low water potential, a little bit of water. Have a look at this diagram that we show you over here. What is moving? The blue. Look at the blue. Okay, so the green is the sugar. So where will water move? If you're having a look here, you will see that water moves from a high concentration to a low concentration. So I've got a cell over here. I've got 2 parts over there. Okay, and what happens is I've got a high water potential on this side. I've got lots of water. And I've got little water on that side. Where will the water move? From a high concentration to a low concentration. And what will it cross? It will cross a cell membrane. Okay, that is really important. A nice concept that you need to be able to understand. Guys, also now the last thing that we're going to look at is sometimes, all right, things I'm going to have to push something up the hill. Okay, so when it comes to osmosis and diffusion, high concentration to low concentration. But sometimes in our body, we actually have to go against the concentration gradient. And I'm going to have to push somebody up the hill. What do I need? I need energy. All right, I need energy to do that. And that is called active transport. Sometimes I'm going to go across, all right, the barrier, where I'm going to go across from a low concentration to a high concentration. What am I going to have a lot of there? Mitochondria. Because why? They need to supply me with energy. Okay, guys, I'm afraid that's all we have time for now. We're going to take a quick break and we'll be right back. Welcome back, Life Sciences, from your break. All right, as I said, let the oxygen going, get the blood flowing. Let's quickly recap. We're looking at this concept of a cell. And we look at the concept of a cell, we can see that there's a structure that has a boundary around it, okay? And we looked at what was filled on the inside, the cytoplasm. Now, all living cells have this boundary called the cell membrane. And we saw that it was fluid, and it's selectively permeable what can go in and out of a cell. Now, sometimes, all right, we find that plant cells, well, actually all the time, plant cells have two boundaries, and we're going to have a look at that concept quickly. All right. So now what I've drawn in over here, something you're going to become quite familiar with, is I've drawn, this is a diagram of a plant cell. Now you can see I've circled, there's a little purple circle over here. Now as you can see on the inside over here, this is going to be my cell membrane. But when it comes to a plant cell, they have got another structure, all right, on the outside, and that is called a cell wall. Now, this is really important as it's only found in plant cells. Animal cells do not have it. So animal cells are just going to have a cell membrane. Plant cells have got this structure called a cell wall on the outside. Now, when you come to why it's important that plant cells have the structure, when we look at the structure now, I want you to think of the following. It is hard, and it is rigid, and it's got like a fixed shape to it. Now, if I were to ask you to go and cut out a plant and open it up, and do you find a skeleton, do you find a support system? The answer is no. So a plant, in order to be strong, to stand up straight, all right, to be able to put the leaves out for photosynthesis to occur, a plant needs to have its own skeleton. Now, it doesn't have that. We have the bones of our skeleton as our support structure. What plant cells have is the cell wall. Now, this cell wall is rigid. It's strong. It's not like the cell membrane that can move, right? It's actually a non-living structure. Now, when I talk about the cell wall, you don't have to know all of this, but its structure is made up of these, all right, if I can say, non-living materials. Now, most of it is made up of something that is called cellulose. There are also two other parts to it, pectin and something called lignin. And very simply, what you need to understand is these parts are hard, all right? They are solid and they are non-living. Cellulose, guys, when you eat it, that is when you eat something, we say you need fiber. What is fiber? Our body can't digest cellulose. So when we eat any kind of plant material, cellulose goes through our body and scrapes our body clean. And it's really important to keep us nice and healthy. But we can't digest it. We don't have an enzyme for it. Right? But the cell wall, in its rigidity, it will allow everything to come through if it could. Right? It's non-living. It can't decide what to let in and what to let out. That's why right underneath it is going to be, you'll see the little yellow and green is going, and that plasma membrane is another word for the cell membrane. So the cell wall, quite intricate with all these hardened substances. And the reason, as we say, if we look at the function just now, why it's there. Now, plant cells need to communicate. it would be very difficult to communicate if I put two bricks next to each other. They are too hard, right? Nothing will be able to move through. But what I do is, instead of it making this solid, I'm going to bring the little razor out here. I have, all right, let me do that. I make these little gaps. Can you see that over here? I put these little gaps in the cell wall, and they are called plasmodesmata. And what the plasmodesmata do, can you see? They allow the cells to communicate. So the cell membrane will still decide what comes in and out of the cell, because the cell membrane is still in between it. But all I do is I don't have the solid cell wall in between these. And you can imagine they're like cytoplasmic strands that stream across from one cell to the other. Because cells need to be in communication with each other. They need to be able to transport things. Now, what is the function? As we said, what is the function of the cell wall? Because it has this solid feature, it supports. And in supporting, it also protects a plant against mechanical injury when it bends in the wind or things like that. So it just gives it that really strong, all right, part. I don't know if you guys use cotton wool. Right, cotton wool is cell walls, all right? Those are all the dead parts of the cell that we're going to use. So only plant cells are going to have a cell wall, but animal cells and plant cells are going to have a cell membrane. Okay, guys, now we're going to look at the last concept in our cells, and this is what an organelle is. Now, very often you get very confused with the word organ and organelle, and basically what an organelle means is a tiny organ. But what an organelle is if I look at here, I have my cell membrane, my boundary, okay? Then I have my jelly inside it, that is my cytoplasm. And then inside the cell, I've got a variety of structures, and we're going to look now, right, at the structure called the nucleus. There are a variety of structures that are going to have a specific function within a cell. So what an organelle is that when we look at this concept of a eukaryotic and a prokaryotic cell, what we noticed is, as I said to you, when a eukaryotic cell became more complex, so it had more things inside the cell that had to do something. So inside the cell, all right, I've got this, all right, these structures, and these structures are all very important, okay? They are surrounded by their own membrane. Now, what is the membrane? It's the same as the cell membrane. So each of these are what we call membrane-bound structures. Now, the most important of all of these organelles. All living cells, all right, all living cells, eukaryotic living cells, that is very important, because a prokaryotic cell does not have this concept of what is a nucleus. So we've looked at what a cell membrane is, all right, so separating the outside and the inside. We have this jelly-like gel keeping everything in space, some hanging around, some dissolved. Now we have this concept of what a nucleus is. Now, when we have a look at what the structure of the nucleus is, this is what the nucleus is. And what the nucleus is, you could say it's the government. Guys, this controls the whole cell. Everything that happens in the cell, everything that the cell does is going to be controlled by this organelle called the nucleus. Okay, so if I was to look at the structure, first of all, I separate it. It's separated from the cytoplasm by its own nuclear membrane. Now, if you have a look, all right, you will see that the nuclear membrane is not solid. it has got these little pores in. And I'm going to explain to you why we need to have the holes there. Now, usually in the center, we've got a very dark staining body. That's when we look at the cells, it's what we see most often. Don't get confused with the words. It's called the nucleolus, right, which is different to the nucleus. Now, the cytoplasm in the nucleus is called the nucleoplasm. And it's the same as the cytoplasm, because this over here would be the cytoplasm. But because it's in its own organelle, its own structure, we call it the nucleoplasm. Now, when it comes to the nucleus, what is the most interesting part? Guys, this over here. Now, what is that? That is called the chromatin network. Now, what is the chromatin network? I'm sure you've heard, all right, you've done the nucleic acids, deoxyribose nucleic acid. And you are very familiar with this twist of what the DNA is. But when the DNA is in the cell, it's 2 meters. It's 2 meters long. It's huge. So what it does is it coils up, all right, and it tangles up, and when the cell is at rest, we call it the chromatin network. It's the DNA. Here, this chromatin network is all of your hereditary characteristics. This is where your genes, who you are, what you are, and this inside the nucleus, all right, is all the blueprint of what you are, what your cell must do. And as I said, it's wrapped around in this bundle with all different things called the chromatin network. And that is why we have pores, because this little guy has to send messages how the cell must do things. It's too big to move out. So we're going to need to use those pores. Okay. As I said to you, the nucleus controls all the activities of the body. It's the most important organelle. It's what tells a cell what to do, what to become, right? And we're going to see all those things when we look at tissues, etc. Okay, guys, I'm afraid that's all we have time for today. Until next time, cheerio, bye.